Good morning, everyone, and thank you for joining today's session. My name is Chun-Shin Lin, Deputy General Manager of Milestone Pharma. Today we are going to walk through the ICH Q3E Guideline together, the new global framework for extractables and leachables. Over the next 50 minutes, I will not just read the clauses to you. My goal is to explain the science behind them, why the regulators wrote each requirement, what problem it solves, and how you can apply it in your daily work. Whether you come from analytical chemistry, toxicology, formulation, or regulatory affairs, you should leave with a clear mental map of this guideline. Let's begin. Here is our roadmap. We have 6 parts. Part 1 sets the stage, what Q3E is and where it sits in the ICH family. Part 2 covers the risk assessment and control framework, the decision logic that determines how much work you actually need to do. Part 3 deals with special situations, cumulative risk, frozen products, single-use systems, and so on. Part 4 is the experimental heart, extractable studies, leachable studies, and simulation studies. Part 5 is where most people struggle, the AET system and the safety evaluation thresholds. We will slow down there and work through the math together. And Part 6 closes with route-specific requirements, ophthalmic, CNS, transdermal, inhalation, plus life cycle management and what your dossier needs to contain. Each part builds on the previous one, so by the end, the whole system should feel like one coherent story. Let's start with Part 1, the overview and scope of ICH Q3E. Before we touch any technical detail, we need to understand why this guideline exists at all and what philosophy sits underneath it. That philosophy will explain almost every specific rule we meet later. So, what is Q3E? On August 1, 2025, ICH published the draft of Q3E, and this is genuinely historic because it is the first globally harmonized framework for extractables and leachables. Before Q3E, if you asked the FDA, the EMA, and the NMPA how to do an E&L study, you would get 3 different answers, plus a patchwork of USP chapters and PQRI recommendations. Companies doing global submissions had to design studies that satisfied everyone, often by doing the strictest version of everything. That is what I call fragmented testing. Now look at the family tree. Q3A and Q3B handle organic impurities. Q3C handles residual solvents. Q3D handles elemental impurities. N7 handles mutagenic impurities. Notice the pattern. Every class of unwanted chemical in a drug product got its own guideline, except the chemicals that migrate in from packaging and manufacturing materials. Q3E fills that gap. It is the missing piece of the impurity control puzzle. 2 more things deserve attention. First, Q3E is the first ICH quality guideline to explicitly embrace NAMs, new approach methodologies, meaning computational and in vitro tools instead of animal testing. Second, its core philosophy, compatibility is not proven by testing everything, it is proven by understanding your risks. Keep that idea in mind, it explains everything that follows. Scope sounds boring, but getting it wrong is expensive. Let's be precise. Q3E applies to new drug products, and note this carefully, including cell and gene therapy products and drug-device combination products. CGT inclusion is a first. Think about why. A cell therapy is a living product in a complex medium, often stored frozen in plastic bags. Traditional E&L thinking was built for small molecules in vials. CGT needs a tailored strategy, and Q3E finally acknowledges that. Equally important is what is excluded: herbal products, clinical stage investigational products, radiopharmaceuticals, and standalone IVDs. Why exclude clinical stage products? Because risk assessment must be proportionate. Early phase products have limited exposure, and over-testing would slow down development without protecting patients. 2 more points on this slide. First, change management. If you change a component, a process, or a formulation on a marketed product, you must reevaluate the leachables profile. E&L is not a one-time study you file and forget. Second, the boundary with Q3D. Q3E owns organic leachables. Elemental impurities stay with Q3D. Complementary, not overlapping. When you design a study, check both guidelines so nothing falls through the crack. This slide shows the skeleton of the entire guideline. Q3E is built directly on ICH Q9, Quality Risk Management. If you know Q9, this structure is familiar: risk assessment, risk control, and lifecycle management, 3 phases that form a loop, not a line. Let me translate each phase into plain language. Risk assessment means figure out what could migrate, how much could migrate, and whether that amount matters. Risk control means, if it matters, do something about it, change the material, set a limit, or generate more data. Lifecycle management means keep watching, because things change, suppliers change, processes drift, new toxicology data appears. Now, two pillars hold this structure up. The first is prior knowledge. This is one of my favorite concepts in Q3E. Your material composition data, supplier extractables data, and experience with similar products are legitimate evidence. If you already know a material is safe, you don't need to reprove it from scratch. The second pillar is the multidisciplinary team. E&L sits at the intersection of four fields. A toxicologist interprets the hazard, an analytical chemist measures the exposure, a materials scientist explains why the material behaves as it does, and a formulation scientist knows the product. If any one of them works alone, the assessment will have blind spots. Q3E explicitly recommends this team model, and in my experience, it is the single biggest predictor of a smooth review. Here is the engine of the risk assessment, the multi-factor risk matrix. It has two dimensions, product quality and patient safety, 7 factors in total. On the quality side, 4 factors. 1. Component interaction potential, basically, how intimately does the material touch your product, and for how long. A tank storing bulk solution for 6 months is very different from a tube the product passes through in 2 seconds. 2. Physical-chemical properties, molecular weight, polarity, solubility. These determine whether a migrant prefers to stay in the material or jump into your formulation. 3. Manufacturing and storage conditions. Heat accelerates migration. Sterilization can degrade materials and create new migrants. 4. Formulation leaching propensity. A high pH or organic solvent formulation is like a strong magnet pulling chemicals out of the material. On the safety side, 3 factors. Route of administration. An oral tablet and an intrathecal injection are worlds apart in risk. Patient population. A neonate is not a small adult. And dosing regimen. A drug taken once is different from one taken daily for life. Plus concomitant medications, because exposures add up. Now the payoff, cross these 2 dimensions and you get your workload. High risk means a comprehensive E&L assessment. Low risk may allow a simplified data package. And oral products compliant with food contact rules may even be partially exempted. The matrix turns a vague question, how much testing do I need? Into a structured decision. Let's zoom into the 3-step assessment itself: hazard identification, risk analysis, risk evaluation. Step 1. Hazard identification. You collect everything that tells you what might migrate: material composition, supplier extractables data, prior knowledge, literature. The output is a list of potential leachables, your suspect list. Step 2. Risk analysis. Now you put numbers on the suspects. How much actually migrates? How much reaches the patient? And where is the action threshold, the AET, which we will spend a lot of time on in Part 5. Step 3. Risk evaluation. You weigh the evidence and conclude, acceptable or not acceptable. 4 things make this method work in practice. First, it is iterative. If your extractable study finds something above the AET, you loop back, identify it, quantify it, reevaluate. This is not failure, it is the design. Second, there is a hierarchy of evidence: prior knowledge first, then literature, then supplier data, and finally your own experiments. Don't jump to the lab before exhausting the cheaper evidence. Third, cross-disciplinary work. Remember the 4 experts from the previous slide. Fourth, documentation. In the eyes of a regulator, if it isn't documented, it didn't happen. Every input, every decision, every justification must be written down. This documentation becomes your dossier later. Part 2. The Risk Assessment and Control Framework. Now that you understand the philosophy, we get practical. The key question of this part is, when can you do less, and when must you do more? There are real regulatory shortcuts in Q3E, but each one has conditions. Let's start with the most surprising provision in Q3E, the food contact exemption. Here is the logic. If a piece of manufacturing equipment, say, a stainless steel tank or a silicone gasket, is already compliant with food contact regulations, and the pharmaceutical use is no harsher than the food use, and extractables testing meets the food safety standards, then that food contact compliance may be sufficient. You may not need a separate pharmaceutical E&L study. Why does this make sense? Food contact regulations like the EU 10/2011 framework or FDA's food contact notifications already require migration testing. If milk sitting in a tank for days is safe, and your oral drug contacts the same tank more briefly under milder conditions, the migration risk is comparable or lower. Retesting would add no knowledge. But, and this is a big but, 3 preconditions must all hold: regulatory compliance, similar or lower leaching propensity, and compliant test results. And 3 warnings. 1. This applies only to manufacturing equipment and systems, never to packaging components or drug delivery devices. 2. Talk to your regional agency first to confirm they accept this approach. 3. Document everything: the comparison, the rationale, the data. An undocumented exemption is an audit finding waiting to happen. The flip side: when must you do a full E&L assessment? 3 situations. First, non-oral routes, injectables, ophthalmics, CNS delivery, transdermal, inhalation. The common thread: these routes bypass the body's natural defense barriers. Your gut is a formidable filter. It has acid, enzymes, and a liver right behind it performing first-pass metabolism. A substance you swallow gets screened before it reaches systemic circulation. An intravenous injection skips all of that. So the safety bar must be higher. Second, even an oral product needs a full assessment if its manufacturing equipment does not comply with food contact regulations. Third, high-risk combinations, aggressive materials like PVC, rubber, or adhesives, long contact times or hot processing, and lipophilic formulations, which are particularly good at dissolving organic migrants out of plastics. Look at the table on the right. It condenses the logic into 2 rows. Compliant oral products may qualify for simplified packages. Injection, CNS, and ophthalmic products demand the full works, TTC, QD, AED thresholds, plus special attention to local toxicity and sensitization. When in doubt, ask yourself, how many biological barriers stand between this migrant and the patient? Fewer barriers means more work. Now we arrive at the single most important decision rule in the whole guideline. For manufacturing components, if all extractables are at or below the AET and no Class 1 leachables are detected, then the leachables risk is acceptable. Full stop. No further identification, no further safety evaluation. Let me unpack why this rule is safe. The AET, Analytical Evaluation Threshold, is not an arbitrary line. It is derived from the safety concern threshold, adjusted by an uncertainty factor. If a compound is below the AET, then even in the worst case, the patient's exposure stays below the level that toxicologists worry about. It is like an airport security scanner calibrated so conservatively that anything it ignores is, by definition, harmless. 3 preconditions, though. All extractables below the AET, not most, all. No Class 1 leachables. Those are the known bad actors like nitrosamines that get no free pass regardless of level. And the study must be run under worst-case conditions, otherwise the conclusion doesn't hold. One caveat from the guideline: this judgment must sit inside the overall risk assessment. You need to be sure you've covered every leaching pathway, not just the convenient ones. But what happens when an extractable does exceed the AET? Don't panic. Exceeding the AET is not a failure, it is a trigger. The guideline prescribes a clear 3-step response. Step 1. Confirm the detection above the AET. Step 2. Identify and quantify. Identification means you determine a structure, ideally confirmed against a reference standard with the same structure, or at minimum a surrogate with a similar analytical response, supported by mass spectrometry and NMR evidence. Record the CAS number, chemical name, and structure. Quantitation means you build a proper quantitative method, with defined detection and quantitation limits, and confirm results with reference materials. Step 3. Safety evaluation, comparing the measured level against the applicable safety threshold. Here is the judgment that resolves most cases. If the quantitated level sits below the safety threshold, the TTC or the QD, and there is no visible impact on product quality, the risk is acceptable. So remember the distinction. The AET decides whether you must look closer. The safety threshold decides whether there is a problem. Most compounds that trip the AET turn out to be fine, but you only know that after you identify and quantify them. Now let's talk about the simplified data package, the reward for being low risk. 2 scenarios qualify. Scenario 1. Prior knowledge adequately controls the risk. If your material composition is well characterized, there is a long history of safe use, and literature supports it, you may not need new experimental data at all. Scenario 2. Your study found only a few Class 3 leachables above the AET but below their safety thresholds. Class 3 compounds are the low toxicity ones. Think of common additives like BHT, so a handful of them above the analytical threshold is not alarming. Look at the comparison table. In a simplified package, basic extractable screening suffices. The leachable study may be waived entirely. The safety evaluation leans on prior knowledge and thresholds, and the dossier is an abbreviated report with justification. In a full package, everything is comprehensive. One sentence from the guideline deserves emphasis. Read it on the slide. Even when you qualify for simplification, the guideline recommends consulting your regional regulatory authority to confirm they accept the approach. Simplification is a negotiation with the regulator, not a unilateral decision. Let's close part 2 with the 3 requirements that make a simplified package defensible. First, agency communication. Confirm acceptability before you commit. Second, robust justification. Show the prior knowledge and the overall risk assessment conclusion that supports simplification. Third, documentation. Record the rationale and every piece of supporting evidence. Table A 1.2 gives 3 concrete examples of lower-risk situations. Materials with known composition and extensive safe use history showed patient exposure with a lower-risk route, and extractables far below safety thresholds with no Class 1 compounds. If your product matches one of these pictures, you have a strong case for simplification. I like to summarize it this way: simplification is earned, not claimed. You earn it with evidence, material knowledge, historical data, clean screening results. You defend it with science, a coherent risk assessment story. And you confirm it with the agency, before the submission, not after the deficiency letter arrives. Companies that treat the simplified package as a shortcut usually end up doing the full study anyway, but under time pressure. Companies that treat it as a regulatory strategy save months. Be the second kind. Part 3. Risk Control Strategies and Special Considerations. The general framework is in place. Now we handle the situations that the simple framework doesn't cover, the edge cases that show up constantly in real projects, transformations, cumulative exposure, freezing, single-use systems, and semi-permeable packaging. Here is a subtle but critical point. The extractable study, powerful as it is, has 2 blind spots, and when you hit either of them, you need a leachable study. Blind spot 1, chemical transformation. An extractable study pulls compounds out of the material using solvents. But once those compounds enter your actual formulation, chemistry happens. A migrant might degrade or react with an excipient, and the product of that reaction could be more toxic than the original. The extractable study never sees this, because there is no formulation in the vial, only extraction solvent. Think of it like testing a food ingredient alone versus cooking it in a dish. The chemistry of the dish can create things that weren't in the ingredient. Blind spot 2, analytical limits. Sometimes a peak above the AET simply cannot be identified, the mass spectrum is ambiguous, or quantitation is unreliable. You are stuck. The leachable study solves both problems because it runs under real conditions, real formulation, real packaging, real storage. It captures transformation products directly, and if a leachable never appears above the threshold in the real product, that negative result indirectly tells you the unidentified extractable peak is probably not a concern. This is the pragmatic escape route the guideline offers, but as the text stresses, you must justify in the dossier why identification was impossible. This slide addresses a trap that catches many teams: cumulative risk. Picture a biologics production line. The product touches a storage tank, then tubing, then a filter, then gaskets, then a filling line. You assess each component separately, and each one passes. Each contributes leachables far below the threshold. Everyone is happy. But the patient doesn't receive leachables from one component, the patient receives the sum from all of them. If 5 components each contribute 1/5 of the threshold of the same compound, the final product is at the threshold. The guideline is explicit: the risk of leachables accumulation shall be evaluated. In practice, this means 3 things. First, map every product contact material in your process. People routinely forget gaskets, seals, and O-rings. Second, group your leachables by compound identity. The same antioxidant migrating from 3 different components must be summed. Third, focus on Class 1 and Class 2 leachables, where accumulation has real safety significance. The good news: this is a calculation exercise more than a testing exercise. If you have per component data, the cumulative assessment is largely arithmetic. Sum the contributions, compare against the threshold, and document the reasoning. Now we move upstream, to a place many E&L programs never look: the containers that store your liquid or semi-solid API before it ever becomes a drug product. Why does this matter? 3 reasons. First, contact intimacy. A liquid API sits directly against the container wall, often a plastic drum, a lined tank, or a bag, for months, sometimes years. Compare that with a filling line, where contact lasts seconds. Second, chemistry. Many liquid APIs contain organic solvents or surfactants, exactly the kind of formulation that extracts migrants aggressively. Third, invisibility. Because this happens before final formulation, whatever leaches into the API carries through the entire process and ends up in the patient dose. So the guideline says, your quality risk assessment and control strategy must cover these storage containers where applicable. In practice, list every liquid and semi-solid intermediate in your process, identify its storage container and contact duration, and ask whether the solvent properties of that liquid make leaching likely. If yes, an extractable study on that container is usually warranted. I've seen companies with impeccable packaging studies receive deficiency questions about a 200-liter storage drum nobody had thought about. Don't let that be you. If you work with biologics, this slide is for you. Many protein products and cell therapies are stored frozen, sometimes at -80 degrees, sometimes in liquid nitrogen. Intuitively, people assume frozen means safe because molecular motion nearly stops. The guideline says, not so fast. You must assess the phases before freezing and after thawing as well. Let's walk the 3 phases. Phase 1, pre-freezing. Your product is filled into its container at ambient or refrigerated temperature and may sit there for hours before entering the freezer. This is actually the highest risk phase, full molecular mobility, direct contact, sometimes elevated temperature. Phase 2, the frozen state. Leaching slows dramatically, true, but watch the material itself. Plastics have a glass transition temperature. Below it they become brittle, and stress cracks can form, creating new surfaces and failure modes. Phase 3, post-thaw. Thawing is a temperature shock. Materials expand and contract, which can pump migrants out of the polymer matrix. And if your process involves repeated freeze-thaw cycles, the effects accumulate. For biologics, there is an extra twist. Proteins can interact with leachables. A migrant might bind the protein, alter its conformation, or catalyze aggregation. So the assessment is not only about patient safety, but also about product quality. The takeaway: design your leachables assessment around the actual thermal journey of the product, not just its final storage condition. Single-use systems have taken over bioprocessing, and multi-use stainless steel is still the workhorse elsewhere. Q3E requires a quality risk assessment for both, but the risk profiles are genuinely different, and treating them the same is a mistake. For single-use components, the question is straightforward: is this virgin material, used once, compatible with my product? A standard extractable study answers it. For multi-use components, 3 additional risks appear. First, cleaning. Aggressive cleaning agents, sodium hydroxide, acids, contact the material again and again. They can degrade surfaces, and cleaning agent residues can react with the material to create new leachables that weren't in the original extractables profile. Second, aging. Heat sterilization cycles, mechanical wear, and material fatigue change the polymer over time. The component in its 50th use is not the component you qualified. Third, cumulative contact. A multi-use component accumulates far more contact time than any single-use item. So the QRA for multi-use components must include cleaning validation, a defined maximum number of uses, and extractables data that reflects the use state of the material, not just the new state. Practical tip: ask your supplier for extractables data after simulated aging or generated yourself on components pulled from actual service. Our last special case: semi-permeable materials, and the surprise is that the threat comes from outside the primary package. Materials like LDPE and certain rubber stoppers are semi-permeable. Small molecules can pass through them in both directions. That means volatile chemicals from the environment can migrate inward, into your product. And where do those chemicals come from? The secondary packaging, the carton, the printed label, the adhesive, the foil pouch, even the desiccant. Printing inks contain solvents and photoinitiators. Adhesives contain tackifiers and plasticizers. All of them are volatile or semi-volatile small molecules. This is not theoretical. There are documented cases in the industry where products in LDPE bottles picked up ink components from cartons during long-term warehousing. The classic example is the recall caused by tribromoanisole migrating from wooden pallets through packaging into tablets. So when do you need to assess secondary packaging? For triggers, you use a semi-permeable primary package. The secondary packaging contains potential migrants like inks or adhesives. The product faces long-term storage, or literature flags your material combination. The assessment itself is mostly a reasoning exercise. Identify the volatile candidates, evaluate the barrier, estimate migration, sometimes supported by a targeted volatile organic screen. Cheap to do, expensive to skip. Part 4, Chemical Testing and Evaluation Strategy. We've covered the why and the when. Now the how, the actual laboratory program. We'll walk through extractable studies, leachable studies, and simulation studies, and clarify exactly what each one can and cannot prove. The extractable study is the workhorse of E&L. Its logic is conservative by design. Instead of waiting years to see what migrates under real conditions, we deliberately exaggerate the conditions to force everything out quickly. Worst case has 3 dimensions: longest contact time, highest temperature, highest pressure, plus the smallest batch size, which maximizes the surface area to volume ratio. More surface per unit volume means more migration per unit of product. But a worst case study is only useful if it is also a sound study. 6 characteristics matter. Representativeness. The material you test must be the material you use, same grade, same supplier, ideally same batch. Extraction power. Your extraction solvent should bracket the polarity of your formulation. If your product is a lipophilic solution, water alone won't reveal anything. Sensitivity. The analytical method's quantitation limit must sit below the AET, otherwise you can't prove compounds are below the AET. Comprehensiveness, cover every leaching pathway. Conservatism, always err on the side of harsher conditions. Comparability, design the study so results can later be correlated with leachables data. This pays off enormously, as we'll see on slide 4.7. Get these 6 right, and one extractable study can support your product for years. Get them wrong, and you'll be repeating studies at the worst possible time. Time to meet the villains of our story, Class 1 leachables. These are compounds with clear evidence of high toxicity. 3 categories: mutagens from the ICH M7 cohort of concern, the notorious structural alerts, compounds whose acceptable intake is below 1.5 micrograms per day, and non-mutagens so potent that the generic qualification threshold isn't protective. Names you will recognize: benzopyrene, a powerful carcinogen, nitrosamines, which have caused major drug recalls since 2018, bisphenol A, an endocrine disruptor. The management philosophy is simple and harsh: avoid them. Choose materials that don't contain them. Only if avoidance is truly impossible do you fall back on compound-specific limits and targeted testing. Targeted analysis is a different game from screening. In screening, you cast a wide net. In targeted analysis, you know your enemy and build a method specifically for it. Benzopyrene by HPLC with fluorescence detection, nitrosamines by GCM-SMS, bisphenol A by LCM-SMS. 2 requirements: the method must be validated as a specific method, and its sensitivity must reach below the concern level. A method that can't detect the compound at the level that matters is useless. How do you know which targets to hunt? From materials knowledge. Read the materials composition. If your tubing is PVC, think about plasticizers and vinyl chloride. If you have rubber, think about nitrosamine precursors and accelerator residues. The target list is written in the material itself. Let's demystify semi-quantitative analysis, the pragmatic heart of extractables screening. In a real extractables chromatogram, you may see 50 peaks. You don't have reference standards for most of them. So how do you estimate their concentrations? You use one internal standard and assume every compound responds like it. That's semi-quantitation, fast, cheap, and approximate. But approximation cuts both ways. If compound X responds twice as strongly as your internal standard, you'll underestimate its concentration by half. Enter the uncertainty factor, UF, with a default value of 0.5, the most conservative choice. The UF deflates your estimated concentration. Actually, more precisely, it deflates the threshold you compare against. Look at the formula: AET equals SCT times UF divided by daily dose. With an SCT of 1.5 micrograms per day and a UF of 0.5, your AET becomes 0.75. The stricter threshold compensates for the uncertainty of your measurement. Now the key logic: when a semi-quantitative result exceeds the AET, that does not mean you have a safety problem. It means this compound graduates to the next level. It becomes a target compound for definitive quantitation or for the leachable study. Most graduates turn out innocent. The AET is a screening net designed to catch everything suspicious, knowing that most of what it catches will be released later. This slide is your decision atlas, the 2 tables from Annex 1 of Q3E side by side. Manufacturing components on the left, packaging components on the right. Let me highlight the differences and the common ground. Manufacturing equipment has 2 unique features. First, the food contact pathway we discussed in Part 2. For oil products, food contact compliance with similar risk may waive additional E&L testing entirely. Packaging never gets that waiver. Second, the prior knowledge route is more flexible for equipment, reflecting the fact that process equipment is often standard industrial hardware with long use histories. Packaging has its own distinctive path. If the only findings above the AET are Class 3 leachables below their safety thresholds, a simplified package with justification is possible. Now the common ground, and this is the spine of the whole framework. 1. Whenever extractables exceed the AET, both pathways converge on a full assessment: identify, quantify, evaluate safety. No exceptions. 2. Both pathways expect you to talk to the regulator when you take a simplified route. 3. Everything below the AET with no Class 1 findings lands at risk acceptable, documented, done. If you remember one picture from today, remember these 2 tables. 90% of practical E&L decisions are somewhere in them. Let's talk about what definitive identification really means, because this is where many study reports fall short. When a semi-quantitative peak exceeds the AET, the guideline does not accept a tentative library match, a software suggestion that your peak might be something. It demands definitive identification, confirmation against a structurally consistent reference standard, using combined evidence, retention time, mass spectrum, and where needed, NMR. Why so strict? Because everything downstream depends on identity. The toxicological assessment of a compound is only as good as your certainty about what the compound is. Misidentify a plasticizer as a similar-looking but more toxic isomer, and your entire safety evaluation is built on sand. In practice, identification is detective work. You start with the library match as a hypothesis. You check plausibility against the material's known composition. Does the supplier's formulation support this candidate? You acquire or synthesize the reference standard, and you compare retention time and spectrum head-to-head. If they match, you have identification. If you truly cannot identify the compound, and sometimes you can't, the guideline requires you to explain why in the dossier and document your alternative strategy, usually a leachable study as we discussed earlier. And remember the quantitative side. Your method must be confirmed with reference materials, with verified LOD, LOQ, precision, and accuracy, traceable to standards. Regulators read this section carefully. Now the gold standard, the leachable study. If the extractable study is a stress test, the leachable study is the real marathon, your actual product, in its actual packaging, under actual storage conditions, over time. 5 requirements define a proper study. 1. Multiple time points. A single end-of-shelf life measurement is not enough, because leachables grow over time and the trend tells you whether the curve is still rising or has plateaued. Typical design, time 0, an early point like 6 months, a later point like 12 months, and end-of-shelf life. 2. The marketed packaging system, the exact container closure and delivery device you will sell, not a prototype. 3. Representative batches, registration stability batches, or development batches if needed. 4. If you can't run multiple batches, provide a justified alternative. 5. Fully validated methods for your target leachables, sensitivity, specificity, accuracy, precision. Why go through all this when the extractable study was so thorough? Because only the real system shows real behavior, the interaction between migrants and your formulation, the actual migration rate at storage temperature, the transformation products that only appear in the real matrix. The leachable study is what the patient actually experiences. Everything else is a model of it. This slide hides the biggest cost-saving opportunity in the whole guideline, the extractables-leachables correlation. Let me explain why it matters so much. Imagine you've established that leachables are a predictable subset of extractables. That is, whatever leaches into your product was already captured by the extractable study at predictable conversion rates. Once that correlation is proven, the extractable study becomes a surrogate for future changes, a new supplier batch, a minor process tweak. You can run the fast, cheap extractable study instead of repeating a long leachable study. Over a product life cycle, this can save years of stability time and substantial budget. The key technical trick is beautifully simple. Use components from the same batch in both studies. Why? Because batch-to-batch material variability would otherwise contaminate your comparison. If the extractables and leachables profiles differ, you want to know the difference comes from extraction versus real conditions and formulation effects, not from the fact that you happen to test 2 different batches of rubber. The correlation itself is assessed along 3 dimensions: profile consistency, do the same compounds appear in both, conversion rates, what fraction of each extractable actually leaches, and time trends, how leachables levels evolve. Build this once, build it well, and it pays dividends for the entire product life. Our last study type, the simulation study, the problem solver for 2 specific headaches. Headache 1, the LOQ-AET gap. Your analytical method's quantitation limit is above the AET. You literally cannot measure low enough to prove compliance. This happens with very low-dose products or stringent thresholds. The simulation study solves it by switching to a surrogate solvent or intensified conditions that concentrate the migrants, effectively boosting sensitivity to reach the levels you need. Headache 2, the impossible matrix. Your formulation interferes with the analysis, proteins precipitate, excipients coelute, and direct leachables measurement fails. The simulation study strips the matrix down to something analyzable and gives you usable data. Now the honest part, the limits. A simulation study simplifies the matrix, so it cannot capture microformulation interactions. It may miss transformation products unique to the real formulation, and its results are approximations, usually conservative, but approximations nonetheless. The guideline is unambiguous. A simulation study supplements, never replaces, a leachable study. One elegant application, accelerated simulation. Raise the temperature, and leaching kinetics follow the Arrhenius equation, so 3 months of accelerated conditions can approximate 24 months at room temperature. Just stay below temperatures that cause phase changes or atypical degradation, and borrow your acceleration model from ICH Q3E stability practice. Part 5, the AET system and safety evaluation. This is the mathematical heart of Q3E, the part where most confusion lives. We'll go slowly. By the end, you'll be able to calculate an AET yourself and defend every threshold in front of a reviewer. Welcome to the most misunderstood concept in E&L, the AET. Let me start with what it is not. The AET is not a specification limit. It is not a pass-fail line. A batch does not fail because a compound exceeds the AET. So what is it? The AET is an action trigger. Think of it as a smoke detector in your house. When the detector alarms, you don't declare the house burned down, you go and check. Similarly, when a compound exceeds the AET, the guideline says, identify it, quantify it, report it, and evaluate its safety. The AET's job is to make sure nothing escapes your attention. Whether the finding is actually a problem is decided later, against the safety thresholds. The workflow on the slide shows it: detect a compound, compare with the AET, and if above, move to identification, quantitation, and safety evaluation. This design has a practical consequence. The AET can be set flexibly, with scientific justification, different products, different doses, different AETs. But flexibility comes with accountability. You must defend your AET calculation in the dossier. Every number in it, the SCT, the UF, the daily dose, must have a documented basis. In the next 3 slides, I'll show you exactly where each number comes from. The first number in the AET formula is the SCT, the safety concern threshold. Its logic is pure toxicology conservatism. Below the SCT, both mutagenic and non-mutagenic toxicity are considered negligible. It is the line below which even regulators stop worrying. Where does the number come from? This is one of the most impressive pieces of groundwork in Q3E. The guideline developers reviewed the permitted daily exposures of about 330 potential leachables, real compounds, real toxicology data, and took the 5th percentile as the threshold. Meaning, 95% of known leachables have PDEs above the SCT. It is a floor derived from the entire landscape of known leachable toxicology, not from a single worst case. Now the relationship on the slide: SCT equals the minimum of TTC and QD. The TTC, threshold of toxicological concern, protects against cancer risk. For long-term oral exposure, it's 1.5 micrograms per day, a number many of you know from M7. The QD, qualification threshold, covers general, non-cancer toxicity. Taking the minimum of the two means one number guides both endpoints. 2 practical notes: the SCT is route-specific. Oral and parenteral values are derived separately because the routes differ in exposure and vulnerability. And remember, the SCT is the starting point of the AET calculation, not the final limit your product is judged by. The second number in the AET formula is the uncertainty factor, and it exists to fix a dirty secret of analytical chemistry. Semi-quantitation lies to you, systematically. Recall from part 4, in screening, you estimate every unknown peak against a single internal standard. But compounds differ in detector response. If your unknown compound ionizes half as well as the standard, your estimate is half the truth. Without correction, a compound at twice the safety threshold could masquerade as compliant. The UF is the correction. Default value, 0.5, the most conservative choice, applied when you have no compound-specific response data. Its effect, the AET equals SCT times UF divided by daily dose. With SCT of 1.5 and UF of 0.5, the AET becomes 0.75 micrograms per day. Notice the direction: uncertainty makes the threshold stricter, not looser. Only results above this deflated threshold demand identification. Can you use a less brutal UF? Yes, if you have evidence. The slide lists the considerations: detector type, FID and UV respond differently across compound classes, structural similarity between your analytes and the standard, the concentration range, uncertainty grows at low levels, and historical response factor data from your lab. Document whichever UF you choose and why. A UF of 0.5 with no data is always safe. A UF of 0.8 with solid response factor data is also defensible. An arbitrary UF with no rationale is neither. Now we assemble the full formula, the one equation to remember from today: AET equals SCT divided by maximum daily dose times UF. Let me make it intuitive. The SCT is a daily safety budget, the micrograms per day a patient may safely receive. Your product has a maximum daily dose. How many grams of it a patient takes per day? Divide budget by dose and you get a concentration, micrograms of leachable per gram of product. Multiply by the UF to correct for analytical uncertainty. That concentration is your AET, the level in the product above which a compound must be identified. The consequence is profound: the AET is product-specific. A high-dose product, say a 2 gram per day infusion, gets a much lower AET than a low-dose eye drop. Same packaging, different thresholds. This is risk-based thinking in mathematical form. The table shows special scenarios. Intermittent dosing, count only actual dosing days. Multi-day products like a 7-day patch, Divide the total by 7. Filters. Calculate from contact surface area in square centimeters. Rubber stoppers. Per container leaching times containers per day, considering punctures. Pre-filled syringes. Barrel, plunger, and needle assessed together with component weighting. Q3ES Annex 3 gives worked examples for each. I recommend keeping the annex within arm's reach when you write your SOP. We've met Class 1 already, now the full 3-class system. Q3ES triage of the entire chemical universe of leachables. Class 1, the known bad actors, M7 cohort mutagens, compounds with acceptable intakes below 1.5 micrograms per day, non-mutagens too potent for generic thresholds. Rule, avoid them. Any detection above the AET triggers compound-specific assessment. Highest priority, no leniency. Class 2, the gray zone, limited toxicity data, not obviously benign, not known villains. Rule, above the SCT, they give both mutagenicity and general toxicity evaluation. Class 3, the gentle compounds, low systemic toxicity, chronic parenteral PDEs of at least 1 mg per day. Common examples, BHT, the antioxidant, and irucamide, a slip agent. Rule, below 1 mg per day of exposure, no further confirmation needed. Above it, a general toxicity evaluation. Step back and admire the architecture. 3 trigger lines, any detection for Class 1, the SCT for Class 2, 1 mg per day for Class 3. This triage is what makes the whole system scalable. Without it, you'd face the impossible task of full toxicological assessments for every one of the hundreds of compounds in a typical extractables profile. With it, your effort concentrates exactly where the danger is. That 1 mg exemption line alone will save your team enormous work. Most compounds in a typical profile are Class 3. So a leachable has exceeded its threshold. Now the safety evaluation begins, and it runs on 2 parallel tracks. Track 1, mutagenicity, following ICH M7. Is this compound a DNA reactive hazard? Track 2, general toxicity for everything above the QD, organ toxicity, irritation, sensitization. Both tracks are mandatory where applicable. One clean track cannot compensate for the other. The general toxicity workflow is classic. Gather NOAEL or load data from the literature, calculate a PDE using the F1 to F7 adjustment factors, and compare it with the patient's actual exposure. If exposure is below the PDE with margin, you're done. But what if there is no data? For the long tail of obscure leachables, this is the rule, not the exception. Q3E makes history. It is the first ICH quality guideline to formally welcome NOMs, new approach methodologies. The priority ladder is on the slide. First, literature PDEs. Someone may have assessed this compound already. Second, read across. Borrow data from a structural analog with the same mechanism, but you must justify the similarity rigorously. Third, QSAR, computational prediction of toxicity endpoints. Fourth, in vitro models when needed. The regulatory signal is unmistakable. Agencies would rather see a well-reasoned read across than a new animal study. This is the 3R principle: replace, reduce, refine, arriving in pharmaceutical quality. My advice, build this capability now, because NOMs fluency will soon separate fast submissions from slow ones. Part 6, Route-Specific Considerations and Lifecycle Management. Different routes of administration face different local risks, and Q3E tailors its thresholds accordingly: eye, brain, skin, lung. Then we close with the dossier and the 5 change types that keep your assessment alive for the product's entire life. Let's start with the eye. Topical ophthalmic products get a clear numerical line, 20 parts per million. Any leachable above 20 ppm in the product needs a compound-specific risk assessment. The number comes from historical experience, decades of ophthalmic products showing that below this level, local safety issues are rare. But note the distinction for intravitreal injections, products injected directly into the eye. There, every leachable requires a qualitative safety assessment. No threshold, no exceptions. The difference makes anatomical sense. A drop on the cornea meets a tissue that blinks, tears, and renews. An injection inside the eye sits in a closed, delicate chamber next to the retina. What do you assess? Local effects: ocular irritation and sensitization, corneal and conjunctival toxicity. Systemic numbers are secondary here. The eye is a local tolerance story. The table on the slide previews the full landscape of route-specific thresholds: ophthalmic 20 ppm, transdermal 500, subcutaneous 50, and CNS plus inhalation requiring compound-specific evaluation. Notice the range, from 20 to 500 ppm. One size never fits all in toxicology. The route defines the risk, and the risk defines the number. Now the most demanding route in all of drug delivery, the central nervous system. Intracerebral, intrathecal, epidural, routes that put the formulation in direct contact with the brain and spinal cord. Why so strict? 3 reasons, and each alone would suffice. First, tissue vulnerability. Neurons barely regenerate. Damage that a liver or skin would shrug off is, in the CNS, permanent. Second, the data gap. We have very little toxicity data for leachables delivered by CNS routes, and uncertainty always pushes regulation toward conservatism. Third, and this is the striking one, PPB sensitivity. The guideline cites in vitro evidence that certain neurotoxic compounds produce biological effects at parts per billion. That is 3 to 4 orders of magnitude below conventional systemic thresholds. A generic QD simply cannot protect the spinal cord. So the rule, no generic thresholds for CNS. Every extractable and leachable above the AET gets a compound-specific evaluation. The 4-step path, identify everything, review the neurotoxicity literature compound by compound, where data are missing, use read-across from structural analogs, and where still uncertain, in vitro neurotoxicity models, noms again. Practical advice, if you develop an intrathecal product, start the E&L conversation with your agency early. The strategy you choose, target lists, models, acceptance criteria, is much easier to agree on before the data exists than to defend afterward. 2 related routes here, transdermal and subcutaneous, and a beautiful example of how Q3E derives numbers from first principles. Transdermal first, 500 ppm. Where does that number come from? Not from thin air. The starting point is the dermal sensitization threshold, the DST, of 1 microgram per square centimeter per day for high-potency sensitizers. Sensitization is the binding worry for skin products because allergic contact dermatitis is a lifelong, T-cell-mediated memory. Then, using the conversion methodology from ICH Q3E, the CTCL calculation, that surface-based dose converts into a concentration in the product, 500 ppm. It covers both sensitization and irritation. Subcutaneous and intradermal, 50 ppm, exactly 1/10. Why tenfold stricter? Because once you inject past the epidermis, the rules change. Subcutaneous tissue is vascular, so absorption differs. Injected drug lingers in a depot, especially with sustained release or viscous formulations, and the injection sites sit near Langerhans cells, the immune sentinels of the skin. A migrant slowly released next to immune cells is a sensitization setup. The tenfold factor buys an extra safety margin. Practical notes, for patches, assess the adhesive and the backing layer, not just the drug contact surface. And for large area patches, convert exposure by contact area. A 60 square centimeter patch is not a 10 square centimeter patch. The lung is next, and it breaks the pattern of neat numbers. For inhalation, there is no fixed threshold. Why? Because the respiratory tract is exquisitely sensitive to sensitizers and irritants, enormous surface area, rich blood supply, and immunologically hyperalert. A compound can trigger bronchospasm or sensitization at levels far below any generic threshold. So Q3E takes the structural alert approach. Any compound whose structure hints at sensitizing or irritating potential gets evaluated, whatever its concentration. Learn these 4 alerts: isocyanates, the strongest sensitizers in industrial chemistry, nitriles, potential cyanide releasers and irritants, styrene, sensitizer, irritant, and carcinogen suspect, and short-chain aldehydes like formaldehyde, fierce mucosal irritants. If any of these shows up in your extractables profile for an inhalation product, you have work to do regardless of level. This applies across the board: dry powder inhalers, metered-dose inhalers, nebulizer liquids. Then a very different situation: products under ICH 9, anti-cancer drugs. Here the logic relaxes. The SCT is defined by the QD alone, the TTC no longer applies. Why? Because these patients already receive intentionally cytotoxic drugs, the incremental risk from a trace leachable is negligible against the therapeutic reality. But the linkage matters. This relaxation depends on the API's genotoxic profile. If the API itself is not genotoxic, the TTC consideration returns. Risk-benefit thinking, written into a threshold. Everything we've discussed eventually lands in one place, the registration dossier. Section 3.5 tells you what reviewers expect, and it comes in 4 blocks. Block 1, the rationale. Why did you design the study this way? What did the risk assessment conclude? This is the story that frames everything else. Block 2, the complete study reports, extractables and leachables data in full. Block 3, safety evaluations for every substance above the AET, including your risk control strategy. Block 4, method validation, proof that your analytical numbers can be trusted. 3 method parameters deserve special attention because reviewers probe them. LOD, the lowest level you can detect. LOQ, the lowest level you can quantify. Specificity, proof that your peak is the compound you think it is, not an interferent. And the critical rule, Your LOQ must be at or below the AET. If it isn't, your study cannot demonstrate that compounds are below the AET, a logical hole that triggers deficiency letters. Fix it with a better method, or justify and supplement with a simulation study. The overarching principle: the dossier must form a closed evidence chain, risk assessment to study design to data, data to safety evaluation, evaluation to control strategy to change management. Reviewers don't just check your data, they check that the chain has no missing links. A common misconception: the E&L assessment ends at approval. It doesn't, it lives as long as your product does. Section 3.6 lists 5 change categories that trigger reevaluation of the leachables profile. 1. New information, new toxicology data on a leachable, or new regulatory expectations. The nitrosamine crisis was exactly this. Products approved for use suddenly needed reevaluation because the science moved. 2. Formulation changes, composition, pH, solvents, anything that changes leaching propensity. 3. Packaging or device changes, new material, new supplier, new manufacturing process for the component. 4. Process changes, temperature, time, contact conditions in your manufacturing. 5. Exposure changes, higher dose, more frequent dosing, longer treatment. The workflow is a disciplined 4 steps. Identify the change and its scope, assess impact on the existing leachables profile, decide whether new studies are needed, and here your extractables-leachables correlation from Part 4 pays off handsomely, then update the risk assessment and control strategy. Notice what's absent from the trigger list: your anxiety. You don't reevaluate because you're worried. You reevaluate when one of 5 defined events occurs. That clarity is a gift. It makes E&L a manageable, auditable process instead of an open-ended obligation. Build these 5 triggers into your change control system, verbatim, and every change review will automatically ask the right question. Let's bring it all home. 50 minutes ago, Q3E was a pile of acronyms. Now you can see the machine behind them. 4 ideas carry the whole guideline. The AET is the trigger, a smoke detector, calibrated per product, that ensures nothing escapes attention without criminalizing every finding. The SCT, TTC, and QD are the safety foundation, thresholds derived from 330 real compounds, so that safety decisions rest on evidence, not fear. The E&L correlation is the efficiency tool. Prove it once with same batch components, and future changes become fast, cheap extractable studies. And norms are the future. Read across, QSAR, and in vitro models replacing animal testing, with regulators actively encouraging it. For your Monday morning, 5 concrete actions. 1. Write an AET calculation SOP, including how you justify your UF. 2. Build a threshold database, TTC, QD, SCT, route-specific numbers. 3. Develop route-specific assessment SOPs. Skin, lung, CNS each play by different rules. 4. Wire the 5 change triggers into change control. 5. Start building norms capability. The companies that internalize this framework will find Q3E not a burden but a competitive advantage: fewer redundant studies, cleaner dossiers, faster reviews. That is the real promise of this guideline. That concludes today's deep dive into ICH Q3E. Thank you for your attention and your time. If you have questions about extractables and leachables programs, study design, AET calculations, regulatory strategy, my team and I at Milestone Pharma are glad to help. You can reach us through the contact details on screen, or scan the QR codes to follow our WeChat channel and connect with me directly. Thank you, and I wish you every success with your products.